Okay, so thank you very much, Rickson, for being here. The floor is yours. And
Okay,
usually we do about an hour. So let's go
sounds great.
for it.
So I guess you can see my screen.
Yes, all good. We can see your screen. Yes, yes.
Okay, sounds great. So thank you very much for the invitation. Thank you, Judith, for organizing this. Yeah, I hope you guys like. We'll be talking about fNIRS data variability. And as we already just mentioned, if you have any questions, just please go to the chat and I'm happy to transfer them as we go along. Okay. So I guess I'll just start with a very brief introduction, right? So basically, the human brain is remarkable, right? So we know a lot about the brain structures. We know which cells we have in the brain. We know how they are connected. So we have a fairly detailed information about the structure information. However, we don't have as much information about brain function, right? We know that it's highly energy consuming, right? So it carries about 2% of the mass, but it actually spends 20% of the total energy that we consume in the body. So we know it's highly energy demanding, but we don't have as much of a great idea of brain function. One thing that we do know is actually the brain has a unique functional organization. And please keep that in mind, because I'll come back to this point along the whole talk, right? And maybe the reason we don't know much about brain function is what attracts us to do functional neuroimaging in general, right? So I guess most of you are certainly familiar with the idea where we actually have a task or something that we wanted to understand how the brain works. And we actually do this stimulation, we perform this task interleaved with a control period, which most of the times is a baseline period. The subject is not doing anything. And we monitor the brain over time. And then we can compare these two situations somehow. And that leads us to see which regions of the brain are different between the two different periods of time. So we can build what we call typically an activation map that highlights the regions that are different between the two control tasks or the two time periods that we are interested in comparing. Right? We can do that with a lot of different neuroimaging techniques. I guess the most widely known is fMRI. We actually started doing this with PET, and nowadays we can do that with EEG, MEG, and of course we can do that with functional near-infrared spectroscopy, right? So the idea is basically the same. This is a group photo of us being happy when we were in the lab before the pandemics. We were not actually aware of how happy we were, but we still were enjoying our time at the time, right? So the idea is actually you have typically a cap where you actually put fibers in, and some of these fibers can actually bring light. They act as light sources. Some of them act like light detectors. And because when you shine infrared light into tissue, you have this light is absorbed by hemoglobin molecules, and a lot of this light is actually scattered, which is kind of for us a good thing because some of this light will be backscattered back to the surface. And this detected light can be related with hemoglobin concentration. And we know that hemoglobin concentration is expected to change due to brain function. So as the brain is working, right, the cell is working, I'll go through all this process. But at the end of the cascade of events, we have an increase in oxyhemoglobin to supply the demand in the higher metabolism. And at the same time, you have a decrease in the oxyhemoglobin. So by monitoring these hemoglobin concentration changes, you can actually find, and comparing these concentration changes in two different periods, you can actually find the regions where brain is activated, right? And that's actually very good because fNIRS has a lot of features that actually makes the technique attractive for us, right? So the first thing is actually it's a safe technique, right? We don't use ionizing radiation. We don't need exogenous contrast. So we can use this continuously, 24/7, which is really good if you want to monitor patients, for example, in clinical settings at the bedside. The second thing is actually it's practical. I mean, light sources and detectors can be very miniaturized nowadays. So you can actually do portable instrumentation, you can do wearable instrumentation. So that allows us to image the brain in natural settings, as opposed to just image the brain in kind of lab settings, right? Like you can't do that with an fMRI, right? So if you want to see something, how the brain works when you are cycling, for example, you need something that can be portable. The third thing is actually it's relatively insensitive to motion, right? So that allows us to assess a wide population, most remarkably infants and babies, neonates, so which is very hard to actually get imaged in fMRI settings, for example, right? More restrictive environments, unless it's anesthetized, which is what you don't want to do with healthy subjects. The fourth thing is actually it's inexpensive. So that actually plays a role as well, right? So it's not as costly as other techniques. So I guess at least these one or more of these features is what attracts researchers to do fNIRS experiments, right? Or to do fNIRS imaging. So I don't need to convince, I guess, this audience about the benefits of fNIRS, because if you're here, probably you're interested in this. But the way we actually looked at this just tell us that maybe fNIRS is very promising. It's a wonderful technique, right? And that's true until you start digging into the results, right? So everyone who has experience with fNIRS knows that it's very attractive. It's even easy to actually do the experiment itself. For not only you have the data to get a meaningful result, can be a really long and time-consuming learning curve. Right? And there are a lot of few points or few specific things that we need to be aware of before we just starting collecting the data, right? So, and that's mostly what I'll be talking about for the next 40 minutes or so. The first thing is actually, and it's a good thing, is actually fNIRS seems to be very reproducible at the group level. Right? So this is just one example of a study that we did with another group that was interested in infants, right? So it's basically a motor performance in 6-month-olds. So basically we just want to understand, or the partner, my collaboration was interested in understanding how motor development worked in infants, and specifically the difference between full terms and preterm babies. Right? So we collected a lot of motor performance data in 6-month-old infants. Right? It's a total of 26 full terms and 25 preterms. That's after discarding probably half of the sample. Right? Imaging infants is actually really tricky. And this is just raw data from the raw group data, right? So you can actually see you have a kind of robust hemodynamic response in the motor cortex in both babies, right? In both the full-term groups and in the preterm groups. So you see that there's an increase around the motor cortex in both groups. You see that some channels in the ipsilateral side, there was a kind of right-hand stimulation. So you see that the ipsilateral side also has some channels that actually respond. If you look at the time courses of the group, you see that there's a robust increase in HBO and a decrease in HBI, right? So it's what exactly...
Rickson, sorry, can I ask you a question?
Sure.
Sorry, yeah, can you hear me?
Yeah.
Yeah, just a quick clarification question. Were the preterm babies 6 months corrected or 6 months after birth?
They were 6 months corrected.
Okay, thank you.
Okay, yeah, they were 6 months corrected. So
thank you.
So yeah, it's a kind of it's a group response. We have seen this pattern actually matches the literature, so it's not really unexpected. But one thing actually caught our attention. This is a group response across 25, 26 babies. If you actually see every single baby and compare the response of every single baby with the group average response, and we did that, what we realized is actually zero. Zero out of the 21, or out of the 51 infants showed the same group pattern of activation, right? So if you see which channels are actually really activated in the group and compare, right? So let's see if this response that we see for the group is the same for every single baby. We see that 0 babies have the same similar pattern that we saw for each respective group. So, and they would actually still see kind of a response in the motor cortex sometimes. Sometimes you see the bilateral response, sometimes you don't see the bilateral response, but you don't see the same profile with all the channel distribution for every single infant. So that actually caught a lot of our eyes, because the group average is actually saying that it didn't really represent the subjects, right? So it actually created an illusion of knowledge, in the sense that we assume typically that the group response is the response, or kind of the robust response for every subject in the group, which was not true. And this is actually not true in a lot of systems. This is only be true if you have a system that we call it an ergodic system, right? Which means that basically, if this is only be true, the group response will be the kind of individual response only if you assume that every member of the group is identical, and if you assume that every member of the group will remain the same in the future. And none of these conditions are true if you're dealing with neuroscience. This is true if you are actually natural sciences, right? So if you are analyzing molecules in a gas, so assuming that every molecule is the same, yes, that's good, and then you can think of the average and drive results of the average, or from the average to the individual. But this is not true if if you expect that every individual in the group can actually change. So, and for this reason, we actually stopped doing group averages, and we are actually starting to report frequency maps. So the idea behind is basically, I do believe that the frequency maps are more informative than group average, because it tells you along the group how many subjects actually have the same response, or have that channel activated, that source detector, or that region, that brain region activated as brain maps, right? So this is actually, in my opinion, more informative, and you can do that as a topography with like we've been seeing here, or you can do that, you can transform this topography and just, for example, show the areas that activated in more than half of your sample for the group, right? And then you actually have more information, or information that's more useful about the group, about the individuals in the group, than the group itself, right? So in this example, we are just showing the same data, but it's a frequency maps of the babies and the regions that were activated in at least half or more of the babies, right? And then you can start seeing that, yes, although you can see there is a bilateral activation in the group, you see that most of the full terms don't have the ipsilateral activation, right? But most of the preterms do have an ipsilateral activation there. So I guess this frequency activation is more informative than any t-value or t-test or p-value that you derive from the t-test when you talk about the groups. Because then I can say, well, most of the groups, most of the preterms, they have activations in one region and the others. And most of the full terms don't have, right? So this tells something about the individuals in the group and not just the group average, right? And the reason there is a lot of this changes, right? From if every individual was the same from the group, so the frequency map would match the group average. But because we don't have the same, we have a lot of variability in the data, we actually can arrive at different conclusions if you see different maps or how you derive the different maps, right? So this is all related to the fNIRS variability, and that's one reason per se why we should care about fNIRS variability, right? So, but there are more reasons why we should care about variability in fNIRS, right? And there's from the kind of philosophical and ethical questions in science, right? So first of all, fNIRS variability is related to fNIRS reproducibility. So if you have a high variability in the data, you have low reproducibility, right? And at least to some extent, we all should agree that science should be reproducible, at least to some extent, right? So this is one already good reason that we should investigate NIRS variability. But there are a lot of others, right? So one of the things is actually more kind of also a kind of, right, maybe philosophical question, right, which is related to group averages. So is actually group average enough in neuroscience? Everything that we want to do, maybe is group average enough, right? So comparing groups of people and see how their brains are different in the group, knowing that this doesn't really match all the subjects that you have in that group, is that enough, right? So are we always be restricted to averages, right, to reporting averages? I know that averages have their place, and I do believe that they do have their place in science, right? So, and this is the same example that I just that I'm giving you is one example that they have a place, right? So in this sense, in this study specifically, we were trying to compare preterms and full terms, right? And we were trying to compare their response at 6 months old and then at 12 month olds, 1 year olds. So you actually can compare, and you can actually say, well, you have two different groups of, in this case, neonates, right, or infants, and you want to, the average can provide you information there at the same time. But I mean, from the moment that you want to say, is this infant really delayed in development or in motor development, or is that infant delayed, or any preterm infant will be delayed or will have an ipsilateral response? So averages are no longer useful, right? So it does have a place in science, but I guess we need more than just reporting group averages, right? We need to be able to actually look at the individual and drive responses to the individual, because remember the first thing that I said, the brain has a unique functional organization, right? So we should be able to actually pay attention to that as well. And that's another reason for why we should care about variability. Sometimes we want to really do protocols that actually look at individual monitoring or also longitudinal studies, right? So these are just two examples that we are very interested in our lab in doing, and the variability actually limits our conclusions, right? One is clinical monitoring. So we want to be able to clinical patients, to monitor patients in the clinic with a very specific disease, which is carotid stenosis, right? Which is just an occlusion in the brain, in the carotid artery that brings blood to the brain. And these patients go under surgery, and we want to compare their vasoreactivity response pre-surgery and post-surgery, right? So in this case, it's a major change. So yes, although there is a lot of variability, we can, at the individual basis, compare the pre and the post. But for example, we cannot compare the recovery over time of these patients, because then the changes are not as sudden or not as deep. And in this sense, the variability actually is a problem, right? So because we don't know whether the change we are seeing is due to the natural variability in the data, or is really related to the due to the procedure or the plasticity, the cerebral plasticity that actually happened during this period. Right? So and the group average doesn't help here, because I'm interested in every single subject and how every single subject responds. and how every single subject responds to the surgery, right? And there are a lot of different experimental protocols where you are actually interested in comparing changes over time, and you are actually interested in looking at the individual basis and not at a group. You're not interested in comparing groups. So another study, right, that has become very popular in fNIRS community is neurofeedback training, right? So you use the brain signal to feedback and therefore modulate brain function. So by looking at how the brain responds to, for example, attention, you can actually show that and try to use this information to modulate attention over time with the brain, right? So this is something that it's very specific to any individual, and the group average won't help much here as well. Right? So, and it's still, you go for a lot of training sessions during neurofeedback training sessions. So you want to be able to compare over time. It's a longitudinal study as well. So these are actually just some very few examples that illustrates exactly why we should care about variability. Otherwise, we'll just stuck or get stuck with group averages forever. And I feel like there's more we want to be able to do with neuroscience, right? And even nowadays that are very exciting. We have exciting opportunities to measure the brain under natural settings, right? And things like that. So really, I feel like we are putting ourselves in experimental protocols where group averages won't be enough to analyze the data. So that actually kind of drove us to really understand the origins of this variability. And because if we can understand where this variability is coming from, we can try to reduce it and have a more reproducible fNIRS result at the subject level, not at the group level. So that's something that we spend a lot of time, and we have spent a lot of time in our lab trying to understand, right? So the first thing is actually, we need to understand where the fNIRS signal is coming from, and that's already explains part of this variability that we see in fNIRS data, right? So we typically, just to summarize, so every time we have neurons working, right, we have a cellular activity at the brain that will actually demand brain metabolism or a higher brain metabolism to restore the baseline of the cellular activity. And that means that we need glucose and oxygen. Therefore, if you have an increase in cellular activity, you have an increase in brain metabolism. And this increase in brain metabolism will decrease blood oxygenation, right? Because it's the oxygen that comes from the blood, right, that diffuses to the cells to supply the metabolism. This is one part of where the fNIRS signal comes from. However, the other part is actually this same cellular activity and the higher brain metabolism increases the demand for blood flow. And if you have more flow going in, you have an increase in blood oxygenation because you have more oxyhemoglobin molecules going there. So, and you still have the baseline flow that actually can regulate the amount of increase in blood flow, right, in local blood flow. So in the end, and whatever we measure, right, so what we are measuring in this cascade of events is at the end of this cascade, right? So the NIRS measurements, the BOD measurements, is typically measuring the end result of this competitive effect of blood oxygenation, right? So metabolism drives blood oxygenation to decrease, and flow drives blood oxygenation to increase. And because there is a mismatch in this increase and decrease, right? Flow increases more than what it's needed from metabolism or what's consumed by metabolism. You have an increase in blood oxygenation. That's why we see an increase in oxyhemoglobin, right? And therefore you have a relative decrease in the oxyhemoglobin there. So this is actually the very end. What we are measuring is actually the very end result of a competitive effect And that per se will actually can vary a lot across different subjects. How we actually process this during brain function can vary a lot, right? And this is not specific to fNIRS. If you look at both fMRI, the both effect is actually the same. And not surprisingly, we see this variation in both as well, right? So this is one result from almost 20 years ago, right, from Miller in I guess, California, University of California. And they actually did a very interesting study. It was an fMRI, it was a cognitive task under fMRI. It was an episodic retrieval task. So the subjects were required to memorize some words, and then they were actually tested to retrieve these words under fMRI. And they see the group average, which sees a significant increase around the left hemisphere, right, more anterior. But when they looked at the 9 subjects that performed the task, they saw significant voxels in completely different places. And the correlation across different subjects was barely none. Right? So basically, this inter-subject variability that I just reported for infants, it's also been seen in fMRI for quite some time, right? And specifically, this variability is greater in situations where we do expect the different subjects to perform different, right? And this is mostly kind of superior cognitive tasks, right? And situations like during development, like the one that I presented with fNIRS in the infants. And this is a population, right? There is a lot of covariates that play a role in this. So every single situations like this, you do expect that there is a variability in brain function across different subjects, right? Just because the way or where your signal is primarily coming from, and this competitive result from metabolism and flow. So this is something that we need to always keep in mind before we interpret group average data and we derive these group average results to individual subjects in a group, right? So as I just showed right from fMRI, it's not just because we have a group activation in kind of prefrontal left hemisphere that all subjects use that hemisphere. Some subjects don't use that hemisphere at all to do the cognitive task here. Right? So this is something that and sometimes if you want to do some or use this information to something, right? It's better to use the information from the individual subject and not from the group average. Right? But there is another problem. Even if you assume that in some situations, the inter-subject, the variability across subjects will actually happen, right? If you get the same subject, right? What happens if you get the same subject to do the same task under different situations, under different times, different periods of time, right? So then we are not talking about different subjects, but we're talking about the same subject doing the same thing, right? It's the same brain performing the same task in different time periods, right? And we did that too. So we did that with the kind of simplest task that we can imagine, since we avoid this cognitive tasks for infants during development. So we went to healthy adults, right, in a very kind of a narrow age range to do a very simple task, which is finger tapping, right? And if you do that, even with very few subjects, in this case, it was 5 subjects, you can actually have a very robust motor cortex response due to finger tapping, right? So you don't even need to go to 20, 30 subjects to see a robust group response. In this case, 5 subjects were enough to see a very robust increase in oxyhemoglobin and decrease in the oxyhemoglobin in the motor cortex, the primary motor cortex region. Right? The problem is, if you get just one of these subjects and you look at their results at 3 completely different days, always perform the same task, the same subject at the same time of the day, but in 3 random days, what you see is actually the response is quite variable, right? And in fact, in this sense, in one of the days, you don't even see a very kind of robust response in anywhere in the brain, right? And in different days, you see that this robust response is different across the brain in different days as well, right? And it can get worse, because even if you get the same subject and do the same experiment 3 times at the same day, in different times of the day, morning, afternoon, and night, what you see is actually, again, a different response. So there is a high variability even for the same single subject performing the same single task. Right. So yeah, there is an despite this across subject variability, there is an intra-subject variability that actually is present in a lot of the data that we see, right? And this is not just specific to the task. So if you do resting state connectivity, right? So the same ideas, but the subject's not performing any task. It's just staying still and quiet for, in this case, it was 6 minutes, right? And you can actually just look and compare the time series, right, of these spontaneous fluctuations in the fNIRS signal. You can compare them quantitatively with computing the correlation, right, coefficient. And if you do that, you can derive, you can find, people have shown that you can find some what's called resting state networks, which are regions that are mostly connected with each other, right? And so yeah, you can see the same in the resting state. And again, if you do group averages of these networks, of these correlations, right? In this case, we did 27 subjects, and we did it simultaneously with both fMRI. So you see the same results, virtually or basically robustly the same results, and it's very robust. And if you actually get one specific region in the sensory motor cortex, you see all the regions that are correlated, temporally correlated with the sensory motor cortex. You can move the time series or the region of the brain that you are interested in, and you can find different regions all the time. So in the top row, each column shows the region we are actually looking at, and each column sees the other regions that are temporally correlated according to the contrast that we were looking at. Right? So you can see, even in resting state, a group average response that is very robust, both in ears and in fMRI. And that's something that we actually have seen in the past. Even we don't need to go with 1/3 of these subjects, right? So I guess over 10 years ago, with very narrow subject cohort, you still see very robust trends for the group averages. But if you just pick one subject at random and look at the 3 different runs that the same subject performed and do the same experiment, you see that the maps, the resting state maps, the resting state networks were completely different at different times for the same subject again. So again, you have a very high variability at the subject level. Right? And part of this is related to, and we have come to learn this, right, over the past few years. So over the past decade, to be more exact, part of this is related to extracortical hemodynamics, right, and systemic physiology. So if you actually look at the brain structure, right, and you are actually shining light from the scalp, right, just above the skin, and basically you want to see what's going on in the cortex, in the most external layer, right, of the cortex. That's where we reach. But the signal or the light that we come from is mostly from regions that do not even probe the brain. So if you are at 3 centimeters, if your source, if your detector is 3 centimeters apart from your source, over 95% of the photons that you detect do not probe the brain, the cortex, right? Just probe extracortical situation. So you are actually interested in just 5% of the photons that you actually detect. All the 95% that you detect, the remaining 95% you detect is not related to the information you are actually interested in seeing, which is cortical information, because these photons don't even probe the cortex, right? And that wouldn't be a problem if we didn't have any interaction, right? If these photons, these 95% of the photons are not absorbed, for example, right? But the problem is that actually, if you see how the hemodynamics of the brain is actually organized and structured, you see that about 60% of the potential for flow control, which means the changes in flow, because the way we control flow in the brain is by changing the amount of flow going into and going out. Over 6% is outside of the cortex, right? It's in the superficial microvasculature, in the pile arteries and the large cerebral arteries, right? So what this happens is actually most of the variation we get and we detect are actually not from the brain, which is what we were mostly interested in, right? And indeed, this extracortical contamination, right, is present all the time. So this is another kind of old result that we have been seeing. We were interested in characterizing the brain. We were not interested in variability, but we saw that, for example, if you don't remove extracortical contributions, if you, in this case, we were interested in arterial pressure and blood pressure, because blood pressure is a way that is a systemic way that we can actually control blood flow, right? So if you collect blood pressure information independently and use this information to regress blood pressure oscillations from the NIRS oscillation, you decrease the amount of correlation and your results more stable at the intra-subject level, right? And this is for the resting state. Other people actually show that for different situations. This is in a very interesting study from Miriam, I guess. She probably talked about this in a previous educational tutorial, right? And the effect of low frequency oscillations, right, which we call Meyer oscillations, due to kind of low frequency pressure oscillations in the brain, and how it can actually affect the estimation of the response to a task. And so this will actually decrease your certainty. It will increase your variability and will therefore decrease your certainty in estimating the brain response or the fNIRS response due to a task, right? And over time, we actually have come to find different solutions for this as well, right? So there are different approaches that came up in the past years. And this is a very interesting study from Ted Huppert's group, which compared different proposals in the literature to deal with these extracortical contributions to the signal. So we can just start with very basic filtering process of the time series, like bandpass filtering, and go to use short separation measurements, position one detector that is very close to the source, and therefore will not have any information from the cortex. And we can use this extra information that is 100% from extracortical, from origins, right? And use this to regress any extracortical contributions. And this actually seems to work very well to account for these extracortical contributions to the fNIRS signal. Although other results, for example, Felix Schockman and Ursula Wolf's group shows that it's not 100% effective, right? So you still see even if you do short channel separations and you regress extracortical information from short channels, you still have remaining physiology that's not related to at least cortical physiology, right? It's autonomic response of systemic physiology. And you can actually try to remove that by collecting independently systemic physiology, like partial pressure of CO2, heart rate, and blood pressure, and any other physiology that can actually be important in this group, right? So yes, part of this intra-subject variability is related to systemic physiology, because if you repeat the same protocol morning, afternoon, after, and evening, you have a circadian cycle, right? And your systemic physiology is dependent on kind of the time of the day, right? So yes, this part of this variability is related to the systemic physiology that we actually acquire. However, in the data that we did with the finger tapping, we also monitor heart rate, arterial pressure, and we see that we try to compare all this variability, right, found in fNIRS with the variability in heart rate, the variability in arterial pressure for all the subjects and for all the conditions. And what we see is actually, although it can explain part of the NIRS variability, it doesn't explain all fNIRS variability at the intersubject level. So there must be something else other than systemic physiology, right? And that's what we actually started looking at. What else can we actually use to remove systemic physiology and to increase fNIRS reproducibility, or similarly decrease fNIRS variability, right? So one thing that, and it's a very simple, the simplest thing that you can imagine is actually motion artifacts can actually increase variability. So we should be careful, specifically in populations that or tasks that induce motion that would be correlated to the task, right? So this can actually add to the fNIRS variability, right? So this is something that, I mean, we were not the first ones to think about that. That's kind of the simplest things that we can think. So a lot of people propose the different situations to deal with these motion artifacts as well. And we try to investigate this, right? So we did a very simple task, which is reading task. It's a reading aloud task, right? So, and just by the fact that you are speaking when you are actually doing the task is related to you moving the temporalis muscle, which can move the skin and therefore disturb the fNIRS signal, right? And we characterize that. So specifically,
sorry,
when
can I? Sorry.
Go for it.
Sorry.
Yeah.
Sorry, can I interrupt for a second?
Sure.
Was looking at the wrong place and didn't see two. So one question is about, one question is, what person gets better at the task? So when you repeat the same task, you're really not repeating the same thing, but rather the person gets training on it at each repetition. So really you're not measuring the same thing. And then there's a second question.
Okay.
So maybe you answer this one because the second is different.
Oh, okay. So, well, the first question, this is actually a very it's a very important effect that you can have, right? So people can learn the task over time. But that's another reason why we went with a very simple task as finger tapping, because, I mean, if you are getting adults at 18 years old or 20 years old to do a some primitive task like finger tapping, you would not expect that the subject will be better in 3 different days, right? As opposed to 20 days. So I agree that habituation's effect can happen, but the degree of the habituation that you expect from a very simple finger tapping task in 3 random days should be very small. It's definitely more of an issue if you're doing a cognitive task. Like the reading task, that can have a large habituation effect. Even if you're, mainly if you're using the same passages, right? But not with a very motor, simple task. I wouldn't expect that habituation could actually play a role, an important role in this.
Thank you. And the second question also about the intra-subject variability is, but this is something you did talk about, is to what extent is due to the time of day or maybe the setup of the NIRS device itself?
Yeah, so I guess it's related to both, right? So I don't know what you mean by the setup, right? There's a lot of things related to the setup, from the instrumentation itself to the probe positioning, right? So what we realized is, well, the time of the day can actually play a role, and mainly because of this systemic physiology that changes over the time of the day, of the course of the day. And since you're getting 95% or more of light that's more related to this systemic physiology than to the cortical hemodynamics itself, you would expect that this will vary. And although we have ways to deal with this now, right, with short separation measurements, collecting independent physiology, they won't be 100% effective. So that's part of this. The NIRS setup, I mean, we didn't test instrumentation, right? We were always using the same instrument, but we are always doing the same protocol. We are always turning on and turning off the system. So there's not kind of stability effects due to this instrument. But we do have something related to the NIRS setup, which is the probe positioning of the setup. How you actually set up your probe on the subject's head can actually make a difference and contribute to the variability. And I'll probably show this in about 5 minutes or so. Okay.
Thanks.
Any other question? No?
I can't see any for now.
Okay, sounds great. So okay, I'll keep going, and please.
Sure, absolutely. Thank you.
Interrupt me if you have anything else. So as I said, I mean, there must be more than just systemic physiology and the origins of the signal-to-use variability. One part of this is motion artifacts, right? So we try to characterize that for a very specific task, which is a reading task. And the motion of the temporalis muscle as you speak can actually disrupt the fNIRS coupling or the optode coupling between the surface of the skin, right, and your optical fiber. And this is a motion that will always be triggered with the task. So you can't actually separate or isolate the motion due to the task with the task itself, right? Every time you repeat the same task, in this case was reading, you will repeat the same motion, right? And if you do that, you can see that there are, specifically to this task, 2 types of motion, right? One, which is related to the task, which is related to the motion of the jaw, right? As you are moving and you are speaking, and that can induce some disruption in the signal. We are exaggerating here. This was an exaggeration by just the jaw movement, right? And the second thing, it's you can always have kind of an uncoupling between the optode and the skin, right? So that will actually drive a change in baseline of the optical signal, right? And this can affect, although you are only speaking at the front, right, with your mouth, it's the anterior part of the brain, this actually can affect posterior channels as well, posterior regions that you were eventually measuring, right? And the change in baseline can also happen in different places, not only the anterior side where your actual motion is being originated, right? And what this does is actually it decreases the ability to detect the power to detect the change that you are looking at, right? And by decreasing the power to detect the change due to the function or to the task in this case, you are increasing the variability of the subjects and across subjects in both intra-subjects if you repeat the same task again and again, right? So this is a group result across over 50 healthy volunteers. And you can see that you can still find regions, but the t-values are very low. Right? It's very, very low, which means that the p-values are also low. And the power to detect the regions that are related to the reading task is quite low. Right? And I mean, people have suggested different ways to actually look and correct these motion artifacts. So we actually tried a couple of them, and we tried to investigate a couple of these motion artifacts, right? So the 2 kind of most popular ones at that time, at least, were the wavelet correction and spline correction, right? So, and that's what we actually tested, and we see that they are actually very good, but they have different ways of working, and they actually kind of attack different types of motion artifacts, or they attack motion artifacts from a different perspective, right? And what we realized is actually wavelet is very good to reduce this kind of more repetitive tasks, but they are very sensitive. The wavelet technique is very sensitive to sudden bumps in the data, right? To this sudden changes in baseline. So that induces kind of a long-term trend in the data. Spline, on the other hand, can deal very well with these trends, with these bumps, but they don't. It's kind of it gets lost every time you are trying to do something that is repetitive with the task. So what we realize is actually if we combine them, right, with spline and then wavelet in this order, right? Otherwise, you may introduce artifacts instead of correcting them. But if you do first spline, then wavelet, it actually works best to correct motion artifacts, right? So this is something that we realized, and it worked very well for this task. We tried a different algorithm as well, which is something that it's also proposed in the literature called CBSI. It's a correlation-based signal improvement. It didn't work as well, and that's why we didn't correct the data here, right? But this is something that we try to look. And again, this is a kind of frequency map, not a group average, but a frequency map across the 52 healthy subjects. And what we do see is actually there is a higher power to detect changes in the reading task. You can actually find the regions that you expect, vernix area, for example, the temporal lobe. And more than that, if you do the task of reading aloud and you compare with a silent reading task, you see kind of better agreement after correcting for motion than the agreement that you have without correcting for motion or just using wavelet with a long pass, a high pass filter, right? To correct for motion. So yes, it does help and it does improve the variability by combining these two different algorithms, spline and wavelet, to really correct the data. And not only you have the same regions and also the same kind of frequency response for each region, right? But if you compare the temporal response across the subjects, you see that they actually have a good match before the two tasks, right? The reading loud, where you have the motion, and the silent reading, where you don't have the motion, right? So you see that there is a very good temporal correlation, both for oxyhemoglobin and for deoxyhemoglobin in the three main areas that we find activation, right? The temporal, vernix, and a more posterior parietal. So yes, using motion correcting, using motion correction or motion artifact correction algorithms can actually improve, and specifically using a hybrid algorithm can lead to a kind of higher reproducibility of the data, both in the areas of we see activation and in the temporal response of the hemoglobin time courses, right? And the second thing, which is related to the setup NIRS that I mentioned before, we said we actually wanted to look at how the lack of spatial information actually plays, what's the role or how the lack of spatial information affects the NIRS variability, right? So this is something that we typically say with NIRS, right? So it's the price we pay for getting kind of freedom to do experiments where we are in populations that we want. We don't have a very specific and detailed spatial information, right? And that's also true. And we actually investigated, it's a very simple Monte Carlo question, where we just get one original probe, right? One position of the probe, and we just shifted the probe 1 centimeter to the side, right? And when you do that, just looking at the Monte Carlo, you see that you change the sensitivity of the probe to detect to some regions of the brain, right? So, and specifically, you can change the regions that you are more sensitive in one place, right? You can actually remove these high sensitivity regions that you are sensitive by just shifting 1 centimeter the probe, which is kind of very common if you're just getting a cap and putting the probe and removing and putting the probe again, it's very easy to get 1 centimeter off in any direction, right? So when you do that, you actually change your sensitivity profile of the data as well, right? So we said, well, maybe we should be able to track where each uptoad is and try to neural navigate in real time with the probe before we start the experiment, because at least we guarantee that we are, it can be not a good sensitivity, and that's a problem with the experimental setup, but at least it's going to be the same, so that will actually decrease your variability. So we actually went and worked with another group from engineering and to develop this real-time neural navigation system. And the idea behind is we have 3 major pieces of it. I'm not going to go into the details, but basically we need structural information. In our case, we actually scanned, for the validation purposes, we scanned the MRI data for every single subject that we collected, because we really wanted to characterize the variability due to the probe positioning. But I mean, you don't need to collect an MRI data for every single subject. One thing that we were about to do before the pandemic struck was see how kind of an MRI map, like a Collins 27 map, can help, and how the variability would hold with kind of standard atlas for MRI, right? The second part is you need to find landmarks in your data, right? And the way we do that is we have a digitizer, and with this digitizer, we collected, in our case, we found out that 7 landmarks is good enough for a good reconstruction. So we actually find 7 landmarks from both anterior and posterior regions, and some landmarks in between. Right? And we mark this so that we can translate the real space where the subject's head is with the virtual space where the digitizer space is, right? And the third, we use a kind of called rate clustering algorithm to actually have this information in real time. So basically what it does is just back projects all the information that you have as a ray, right? And projects that in a 3D screen. And that's a movie that you can actually see how we see that. So in a very region, you have the digitizer. And as you move the digitizer, you can move the information, which is the red cross, and you can see in both the 3 views, the actual coronal and the 3D volume, where your probe is, right? So we can actually, in real time and very fast, navigate and mark different positions. And of course, we can mark the position of each optode in this data, right? So we actually quantified this, so I'm not going to go into the details, but we see that compared to the MRI information, right, we can do that with a very position of vitamin E, for example, in the head, on the head of the subject, and do the experiment inside the MRI. We see that the digitizer algorithm, right, the neural navigation system, has an error of about 4 millimeters, which is good for us in the sense. Consider that the spatial scales that we use in NIRS, right? And one issue that can come up is the registering the landmarks, so we can be subject sensitive. So we tried 5 different people to do those landmarks and to register the landmarks, and we see the reproducibility within the 2 millimeter region, right? So if you sum the absolute error with the variability due to the mark, you have half centimeter, which is low than what we typically are using. So we wouldn't have enough spatial information to distinguish 5 millimeters in weight. So it was good enough for us. And what we did is actually repeated the same experiment, finger tapping, right? And but we did 2 different protocols. One with a standard approach, where we just use what we were using to do before the neural navigation, right? We would just get the cap and position it. We use the 1020 system to actually mark the regions. And then we use the guided approach, which with the neural navigator. So in this sense, we were interested in a finger tapping task, which is a motor task. So we used an uptoad in the motor cortex as a landmark. So we actually neuronavigate the motor cortex, and we would actually mark the optote that we are interested in, or the source of the detector that we are interested in. And every time we repeated the task and the experimental protocol, we will always navigate to the same region. Right? So the top row is 5 different subjects. Every time you do the standard approach, we repeat the same protocol 3 times at 3 different days. And what you can see is actually the reproducibility is quite low in all the 5, in at least 4 of the 5 subjects, right? So the color you see is the amount of times that the channel was activated in every single session, right? And we repeated it, we have 3 different sessions because we repeated the same protocol at 3 different times, at 3 different days. Right. So one subject to have a high reproducibility with the standard approach, which is probably more related to the size of the response than to the reproducibility of the response itself, because we have a large area of activation there for this subject. With the guided approach, it's the opposite. We could find at least one channel that was reproducible in 4 out of the 5 subjects. So we could actually reproduce the same experiment at 3 different days by just neuronavigating to the region that we were interested in, right? So by doing this, we could actually increase the size of the response, but also the overlap of the response across the 3 different days that we actually collected data from. So just to end now, why we want to do that? Because all these improvements helps us to do the studies that I mentioned in the beginning, right? So this is one example that we're doing in the clinic during the neuro ICU. It's not exactly with fNIRS. We are using DCS at the same time. And we are trying to monitor patients that actually developed brain ischemia due to traumatic brain injuries. And in this case, what you really want to monitor is the ischemia itself. So you don't want to be wrong. You don't want to be 1 centimeter off. You want to really measure the ischemia and what's going on in the ischemia. So by neuronavigating in real time, motion artifact is not a problem in this case because the patients are typically in coma, so they don't move. But if with the neuronavigation tool, you can actually go exactly where you see the ischemia, and you can monitor the region of interest without variability from one day to the other. Sometimes you can, this patient, for example, we monitor for 9 days in a row. So you can actually put the probe on and off every day, and you can actually do look at the same place, and you can understand how the ischemia is developing in a more reproducible way, right? And the other thing is going back to what we said in the beginning, right? So we have a unique functional organization. If that is true, we should be able to recognize by the brain patterns the unique organization of every single individual and not the average group. So this is something that we are very interested in now, using machine learning to identify subjects based on the resting state profile or their response profile, right? So if every individual is unique, as much as they actually see the same, they are not the same. And maybe classification algorithm and machine learning can help us identify subjects. And we can do that with over now about 95 to 97% accuracy. Right. So in summary, I feel like there's still a long road ahead towards monitoring individuals with fNIRS, right? Variability and decreasing variability is part of this, and this is what we have done, right? But I guess over time we learned a couple of things. The first thing is actually fNIRS is really wonderful and really promising. So we should keep using fNIRS. However, we should be aware that fNIRS variability limits some experimental protocols and some data interpretation, right? And we need to be aware of these origins before interpretation of the data. Otherwise, we can misinterpret the data. And motion artifact correction, spatial information can improve both the inter-subject variability and the intra-subject variability. And in the end of this journey, I guess it may be worth the journey, right? And there's a famous saying that life is about the journey and not the final destination. So I just want to end thanking my group and a lot of collaborators. A lot of data that I presented is not from us. We are not trying to solve our problems. We're trying to develop solutions for collaborators to do their studies and do the interpretations in cognitive science, developmental science, and things like that. I want to particularly thank Sergio Novi and Edwin Ferreira, two PhD students that actually did part of a lot of the work that I presented here. Deborah Miranda, who I actually run all these babies studies at in Brazil, and Glenn Telles and Corey Telles in Misericordia University, where we run this reading task in the speech and pathology, speech pathology department. Right. So that's all, and I hope you guys enjoyed, and I'm glad to answer any questions you may have.
So fantastic, thank you very much, Rickson. So as before, feel free, for all the participants, feel free to post your questions in the QA session or in the chat. We had a couple of questions as we went along, so I'm guessing that people asked most of their questions already, but I do see one question in the QA. It says, thanks for the nice talk. What do you think is the next step for improving fNIRS reproducibility? Would it be hardware or stats data analysis related? Thanks.
Yeah, well, I feel like both play a role, right? And I guess we are going towards both directions, right? So in hardware development, I feel like we have come a long way, right? We found that NIRS, as inexpensive as it is, and we like to say that all the time, it's way better if we combine independent measurements, for example, with systemic physiology measurements, if we combine digitizers, for example, to navigate in real time. So there is kind of this hardware improvements that we actually gathering towards a better detailed picture of NIRS, right? And this is one part of this. And by NIRS itself, I guess we are going into high density probes, which eliminates a lot of these problems, right? So if I really want to understand, for example, the motor cortex response or the prefrontal cortex response, by having a high density arrangement, maybe I don't need to worry anymore about where my probe is, because I can do tomography with the data, with the overlapping data that we'll collect, rather than just doing spectroscopy, which is looking at multivariate channels, right? So that actually can be a good, nice improvement, being able to do tomography as much problems as we have with tomography right now, because it's not a unique problem to solve the reconstruction process. But it's, I guess we can be better than just doing spectroscopy, right? So this is one part of this. And that also responds like the stats or data analysis improvement as well, right? So if we really working towards better reconstruction algorithms for high density probes is one statistical or data analysis improvement that we can do. And I guess there's a lot that we can do mainly with now we are in this vibe of machine learning, and it's reasonable in the sense that it has been proven to be very useful in a lot of situations and also in neuroscience. But I wouldn't say that not even machine learning, we can go there. I guess there are a few steps that we can actually improve. And there's a lot of multivariate statistical analysis that we can do and to better understand this relationship. The brain is something that we ignore quite often. The brain is a complex system, and we typically analyze the brain looking at very kind of regions, right? So sometimes we image the prefrontal cortex or we image the motor cortex, and because that's the region we are interested in, and that's understandable, that's where we expect the response. But the nice thing about complex systems is that they are not, you can't split a complex system by parts. So you need to look at that as a whole. And that's why I believe that functional connectivity can help us understand better the brain, because you are looking not only where the regions are activated kind of individually, right, channel by channel, but you can look at how these regions are connected to each other. And I guess that's what the brain or any complex system is about. So I feel like there are a lot of improvements that we can go in both directions, hardware and analysis.
Okay, another question. It is focused on resting state functional connectivity. What do you think about using wavelet filter for motion artifact correction and using wavelet coherence to measure functional connectivity in the same signal processing pipeline? Do you think this can lead to more variability in subject level results as they can interfere with each other?
Yeah, well, that's a good question. I don't know. You are using the same rationale, so it's probable that your first wavelength decomposition will interfere on your wavelength coherence results in the end, right? So that may be an issue as well. It's just suspicions. I never really tried that, but since you're doing the same decomposition, you can, and probably you're using the same kernel function as well. You probably can have interference results there, but yeah, there are different ways you can correct motion artifacts, and in this sense, the best way to correct motion artifacts is not needing to correct it. So trying to avoid this with resting state connectivity, it's easier to avoid motion correction because the task is simpler, right? There's no task. You just have to sit down or and do nothing, right? And ask yourself to do nothing. So you are less susceptible to this kind of sudden, to this regular, because remember, we were using wavelength for motion correction to correct for these regular patterns that are correlated with the motion, with the task. And in resting state connectivity, you are not doing that, right? And wavelength decomposition is not very good to deal with sudden bumps, right? So it introduces long-term changes in the data, which you could principally correct, which we could correct later with kind of high-pass filtering, but it's not as effective as well. We tried that. So what I would say is actually, if you're doing resting state, you probably can skip wavelet decomposition for motion correction artifacts. And spline will be more important because the type of motion that you are subjected to resting state connectivity is more sudden bumps from just spontaneous movements of the head during the period you are collecting the resting state data. And you don't have very repetitive motion from resting state data. So that's what I would say. Probably I would skip the wavelet for motion correction if you are interested in doing the wavelet coherence analysis in resting state activity. Another thing to do is try to do, instead of looking at the frequency spectrum, right, for quantifying the spatial temporal relationships, you can try to look at the temporal and do, I don't know, Pearson correlation coefficients and things like that. There are differences between spectrum and temporal correlations, but that's another approach to do as well. So, but I feel like for resting state connectivity, this is less of a problem in terms of wavelets. So you probably can skip that. So I would probably do.
So thank you very much. I think there are no more questions, and maybe it's a good time to just stop here. Thanks a lot, Rickson, for this really wonderful and very accessible presentation. And for all those of you who are here, as usual, this was recorded and will be shared on the Society's website. And we have one or more tutorials coming up still before the summer break. There is also an answer in the chat for the wavelet issue to your answer. So thank you very much, everybody. Thanks a lot, Rickson, for the talk, and hope to see you at the next tutorials, everybody.